Okay, I'm gonna start. We're gonna get a little bit into DNA. Now, so happens that we're going to be talking about basically kind of a scale between what is made up of what. So if we have a whole organism, whether it's a human or a giraffe or a carrot or a bacteria, all those things are made up of cells, right? And within those cells have functions, depending on what kind of cells they are. And those functions are carried out by proteins. And proteins are made up of, you guys remember what proteins are made of? Amino acids, right. Okay. And those amino acids are coded by DNA. That means coded, in other words, instructions. So here's our different cells. And then each nucleus contains an identical complement of all the chromosomes. And each chromosome, there's 2 copies. There's one copy from mom, one from dad. And then all those chromosomes contain the whole genome, not just the genes that cell needs. Everything. Okay? So that's the instructions, essentially. Except that it's not exactly, it's instructions that are moderated by the environment. Whatever kind of environment we're talking about at a different scale. So it could be the outside environment. It could be the cellular environment, all the other cells around it, or all the cells in the body that are talking to each other. It could be the DNA environment. So I want you to understand that there's different scales of the environment. And then on each chromosome, so here's a specific chromosome pair. These pairs have the same genes on them. Okay? One's from mom, one's from dad. Now, the same genes, but not necessarily the same alleles. So, remember what an allele is? Anybody? An allele? We talked about it before. Yeah. Exactly. So, an allele is a gene variant that codes for a slightly different protein. Now, in some cases, the variant has fixed and there's no other alleles for certain genes, but for lots of them, there could be, in some cases, 1,000 alleles for one gene. And these alleles are passed down from parents to offspring, from ancestral species to descendant species. And every person, because we have 2 copies, so there's a chromosome pair, one from mom, one from dad, that's called diploid. So, 2 copies. So each person in here has 2 alleles. They could be the same alleles, or they could be different alleles. Sometimes you get the same allele that you inherit from your mom as from your pa. Other times, you might get 2 different alleles. And then each chromosome is one long DNA molecule, and genes are the functional regions of this DNA. So there's also parts of this DNA that does not code for a protein or amino acids that make up a protein. There's been a big debate about why there's all this extra DNA, since only genes make up about 2% of our total DNA. And now some of this DNA is actually what we might call junk. It's a lot of it is what's called transposons, which are parasitic segments of DNA that can copy itself from one part of the genome to another. And other parts are old genes that have had a mutation that have basically made them worthless. Now, it turns out that a lot of that stuff, though, isn't junk. Transposons are crucial. Transposable elements is what they're called, are actually crucial for gene regulation. So there's a lot of regions of the chromosome that regulate genes that are on the chromosome. In fact, the genes themselves, while they're important, it's the regulation of those genes that makes this situation so, I would have to say, magnificent. Okay, and the DNA is made up of a double helix, and it's composed of these bases. A and T go together. So A is adenine, T is thymine, and then C and G, cytosine and guanine, always pair. So you never have a situation where you have an A and a C as complements. We'll look at the DNA structure on the next slide. So here we are. Now, the backbone of the DNA is made up of sugars and phosphates. And this is the backbone, right? And then the other side. Okay. And then finally, the bases are these things. And if you notice, let me get to red, that'll be better. Okay. If you notice that there's a pair of bases there. In this case, it's shown as either blue or yellow. But they correspond to, they're either an A and a T together, or a C and a G. And these bases are called nucleotides. Okay. Okay. And here we see it again. So the P's are the phosphates and the sugars, and then the nucleotides are these right there. And a triplet of nucleotides in a row. So we'll read this strand right here. Would read ACG. And ACG corresponds to an amino acid that makes up a protein. So an amino acid is coded by 3 base pairs. There's a little video on Watson and Crick. They discovered the structure of DNA. Now, this was back in the 1950s, early 50s. And at that time, they were just starting to think that maybe DNA was the molecule that contained the genetic material, in other words, the genes, that coded for proteins. But they didn't even know that much. So the question was, what is the hereditary material that we learned about in terms of inheritance from the evolutionary part? And the DNA is actually the inherited material that is then passed on from one generation to the next. And furthermore, when cells actually have to replicate, for example, our skin cells replicate fairly often because old skin cells die and they're replaced by new skin cells. So the only way to replace them is for cells to actually replicate themselves. And that replication, with that last video, you get a new double strand. Each cell has a new complement of chromosomes, and then they can carry out their functions. But anyway, these two guys, one was an American who's still alive, and the other one was British, name is Crick. So Watson and Crick. Now, there was a big race for figuring out the structure of DNA. At the time, they didn't know it was a double helix. Some people actually thought it was a triple helix. Okay? There were other people working with what's called X-ray crystallography to get actually at the molecular structure. And once that information came out, these guys put it together and realized it was a double helix. Now, a double helix means that, so we have these complementary strands and complementary bases. So if you only had one strand, you could make up the other. So let's say you have a strand that's A, C, C, G, A, A. You could come up with the complementary strand. Okay, so the first one was A, so the complement would be T. There were 2 C's, so the 2 complements would be GG. Okay. Now, to actually copy a strand in terms of DNA replication, anybody remember what anything that ends in ASE signifies? Kind of protein. Enzyme. Exactly. So, DNA polymerase is an enzyme that strings together the complementary strand during DNA replication. So, if we start with this double strand, it starts to unwind. Each of these little these are the bases. Okay. Okay. And then each of these separate strands, a new complement corresponding to the other side of the base pairs is made. So now we have two strands, two double helixes, and each of those separate double helix chromosomes, one pair is going to go into remain in the original cell, and then there's a daughter cell that's exactly the same. Now, once in a while there's some errors. Those are mutations. Okay, we already saw this. Okay. So these nucleotides, we have two alleles, one on each chromosome. Now, people who are more related to each other share more alleles than people who are less related to each other. So, offspring or siblings share, we'll say on average, 50% of their DNA. Parents and offspring share 50% of their DNA. So if you compare the bases and you see that, oh, there's very few differences among these two people, we can probably say, oh, they're probably related to each other. They share the same alleles because of common descent from the same ancestor. In this case, their parents. Okay, so we can use this information to actually identify people. This is where DNA analysis comes in. We can use it to identify crime victims, perpetrators, human remains from missing persons or disasters, and we can determine relationships between family members in terms of paternity and missing children and whether they've been abducted or not. Okay? So if you find, let's say, a body and you don't know who it is, you can then get the sequence down and you can submit it to a database to see if it matches someone in the database. Now this is called forensics. In terms of, are there any criminology majors in here? Okay. Now, for the longest time, people were matched to or identified based on their fingerprints. There was also all this stuff about hair analysis and forensics. They used to call it, well, they call it forensic science, but it's not science. It does the opposite of science. They try to prove that the the remains of this person belong to supposedly someone that they can figure out, or the criminal who, let's say, committed the crime did it. Now, DNA analysis is much more scientific. We know how alleles are passed from one generation to the next. We know that the chances of someone sharing exactly the same alleles with someone else are like 1 in 7 billion, something like that. But things like fingerprints and bite marks and all that stuff, it turns out that's worthless stuff. They still use it in criminal courts. It's unfortunate because they're idiots. How's that? Okay, so we can get DNA evidence because we leave behind DNA all over the place. We leave cells behind wherever we go. Whether it's the skin underneath a victim's fingernails, saliva, skin on dirty clothes, the butt of a cigarette, saliva with cells in it. There's all kinds of DNA. In fact, it turns out you'd be surprised where you could try to like clean up after yourself and you would still leave thousands of cells of DNA wherever you were. So right now, you're sitting in your desk, and I'm sure you're leaving cells that we could collect, and we could then show that you sat in that desk, and while you were sitting there, you were murdering someone. Okay? I know it because I could get your DNA. Not that you were murdering, but you know, I have an active imagination. Okay, so now the thing is, if we collect your DNA, these cells, and each nucleus has a full genome, that's just one copy of your DNA. Well, to do an analysis of DNA in terms of what alleles make up a certain person's sequence, we need a lot of copies. And so we have a technique for making millions and millions of copies. It's called polymerase. Now, ACE again, so that's an enzyme. Chain reaction, PCR. It means that we can take a strand of DNA and automatically figure out the complement and then make millions and millions of copies. So then we can then figure out, okay, who does this piece of DNA belong to? Now, one of the beginning is that these DNA, this double strand, you can actually get it to separate by heating the DNA. And the strands separate into separate strands. And then from each strand, you can make a complement to it, just like our cells do during replication. So here's the PCR cycle. You start with a strand of DNA. You make identical strand. Those two separate strands make another copy of themselves. So you got 4, 8. Each cycle, you double the amount of DNA. Now, there are these regions on the DNA that are variable. That means they usually have lots of variation in the alleles that are there. And those variations depend on the individual. So you can that way, because of these variable regions, someone might have a C, AG, and someone else might have an ACT, okay? And so you can actually identify these individuals by their variable regions. Okay. Now, these fragments of DNA are called short tandem repeats. And DNA analysis in terms of forensics focuses on these STRs, short tandem repeats. Now, as I mentioned, you need 3 base pairs for an amino acid. But in this case, we have these repeats, there are actually 4 of them. Doesn't really matter, we just want to be able to identify the individual. And so, in this region, there's 1, 2, 3, 4, 5, 6, 7, 8. There's 8 repeats of the TAGC repeats. Okay? Someone else might have 4 repeats in this region. So they'll go TAGC, 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 TAGC. Another person might have 7. Another person might have 8, like another person. But if we pick a bunch of these different regions across the genome, salute, the chances that an individual will have the same number of repeats at a whole bunch of these different sites as another person becomes exceedingly unlikely. Salute again, Gesundheit. Okay. So we use these things called primers with the PCR. That means they're kind of attached to the sequence that we want to look at, because these primers on either side, we know that they're there, and so we can make primers that attach to that. Now, the primers don't vary between individuals. In other words, the primers are shared so that we can use the same primer for all of this analysis. The only thing that differs is between the primers we have these STRs, short tandem repeats. Okay. So here's our gel that we were briefly introduced in that kind of lousy video. And there's something that you should notice. So this is person 1. And at this one locus, they have the same base pairs because they're identical from mom and pa. Okay? And then this other person, number 2, oops, this person has 2 alleles. Remember, 1 from mom and 1 from pa, at different loci or different places. One is shorter, the short tandem repeats. One is shorter than the other because, as I'll show you in a second, the fragments move through the gel and they move more quickly or slowly depending on how big those fragments are. So, as I mentioned, if a person has, like we saw before, 8 repeats, and from one parent, and 4 repeats from another, we'll say this band will be 4, or this band will be 8, and then this band will be 4. Shorter fragments move more quickly because they weigh less compared to longer fragments. So remember, everyone inherits 2 copies of each short tandem repeat. 1 from mom and 1 from dad. So here's our gel electrophoresis. Okay. We have DNA fragments of different sizes. Okay, we put, for one person, we put DNA samples into the wells and the gel. So there's this gel, and for each individual that we're looking at, we have a different well. Okay, so we might have a reference gel, and then we might have, let's say, suspect number 1. And then suspect number 2, and 3, and 4. Okay? And we put these fragments, one from each individual, into these wells. Then we actually apply what's called an electrical charge. As I mentioned in that little video, DNA has a negative charge. So we apply electrical charge. Remember, an electrical charge is negative. The DNA and the electrical charge repel each other, and the DNA strands and fragments then separate and go towards the positive. Okay? Now, the DNA fragments, depending on the size or the number of base pairs or repeats that each individual has, the fragments move through the gel at different speeds. So, larger fragments move more slowly through the gel, and smaller fragments move more quickly. So if this fragment, let's say, is a repeat with 2 repeats, or rather 8 repeats, it's going to move much more slowly than this one with 2 repeats. So 8 repeats is longer, it weighs more. Okay? So it doesn't move as far. Now, once they move through the gel, we can then assign them. We know that this person here has 2 fragments that are the same length. This is a double fragment. Okay? Let's say it's 5 repeats long. This person here doesn't share either of these fragments with this other person. It's got 1 fragment at 4 and 1 fragment at 7, let's say. Now, person 3, right? It's actually got, it shares 1 fragment with person 1. Okay? Those fragments are identical in length. It could be because they're related to each other, or it could be because it just so happens there can be, let's say you have anywhere from 2 to 8 fragments, 2 to 8 repeats in a fragment. Obviously, just by chance alone, you can end up with an individual with the same size fragment. Again, person 2 and 4, they share this fragment together. And then this is a double fragment with this individual and this individual. Okay. So remember, Smaller STRs move more rapidly from top to bottom than long fragments. Now this is just for one variable region. As I mentioned, we don't really use just one variable region for each person. Originally, they used 13. Because even if you share a fragment that's identical length with someone else, that is, let's say, not related, on another variable region, you will not have probably the same allele or fragment length. You'll probably have a different one. Whereas if the two people that you're looking at are actually related to each other, then they'll share more of these 13 regions with the same allele. Okay, so it says which STR is smaller, the top one or the bottom one? Bottom. Smaller, it moves further. The larger one is on top. Okay. So here we have a problem. How do we figure out who is not the father of this mom? Forget about the mom, but the baby. Okay. Now remember, each individual, so here's mom. She's got a fragment of this length that's longer than a fragment of this length. Okay? The baby shares this allele from mom. Okay? And then there's this one other allele up here. And this allele is from dad. Okay? Okay, so here we are again. We've got mom and baby. And then we've got dad, or rather mom, and the baby, here's the baby's father. Okay, so we're only interested in this fragment up here. Now, if we go across, we've got three different possibilities for fathers. We see that this fragment is identical in length to this person or this person. So both of these could be fathers. We don't know because there's going to be 13 different areas, and if they're fathers, they'll match up. So who is absolutely not the father of this baby? One, right. Okay. Now, it's actually not even the mother of this baby either, as you can see, because here's mom right here, and there should be a band right there, and there's not. Okay. Now, I mentioned that the more short tandem repeats you test, the more different variable regions, the more you can narrow down the possibilities of who's related. Because if you just used one, there's a very good chance that someone might have the same length of that STR at that area, but not because they're related to each other, just by chance alone. But if you have 13, the chances that that individual will share that fragment length at all 13 becomes very, very small. In fact, we don't even use 13 anymore. I think they're out now up to 20. Actually, instead of using fragment lengths now, we actually use those nucleotides in terms of which nucleotide a person has in terms of the 3 base pairs, but don't worry about that for now. So these are all the chromosomes, 1, 2, 3, 4, 5, 6, 7, 8, all the way up to 22 plus the sex chromosomes. And each of these areas, one of their variable regions that they look at is this one on chromosome 2, this one on chromosome 3, this one on chromosome 4, and so on. And then the sex chromosomes, AM, EL, on the X and on the Y. Okay? So here we are again. We're going to try to figure out who the father is. Okay. So here's mom. And this is, baby shares this with mom, this fragment length. Here's mom's other fragment length from, so one's from the mom's mom and one's from the mom's pa. So we're not really concerned about this one. We're concerned about this one. That's going to be the father. And so the mother and father of this baby are going to have this length and then this length. Well, that's mom, but it's going to be over here somewhere. Okay. So again, here's mom. Here's dad. And where's dad? Right here. So this is the father. Now, the human genome contains thousands of STR markers. For a while, the FBI used 13. They now either use, if they use this STR thing, they either use something like 20 or 30, something like that. And, or they use this different thing where they actually identify the nucleotide at each place. So if you use a standard set of markers, allow information to be shared at local, state, and federal. So that's why, let's say you have an individual who has been convicted, let's say 10 years before, of raping this woman. At that time, they didn't have this technology, but they still collected various fluids like semen from the woman during the test. Okay? Well, the guy who they convicted said, I didn't do this. Of course, they didn't believe him because all suspects are obviously guilty or they wouldn't be suspects. Isn't that right? Not really. Some might be guilty and some might not be guilty. So, the poor guy who's been in jail for 10 years for raping this woman, he didn't do it. And he said he hasn't done it, but no one believed him. He got convicted anyway. And finally, his lawyers managed to get a test of the material that was collected at the time, but they didn't have the technology for doing this. And lo and behold, they find out that this poor guy didn't rape this woman because when they tested it, his fragment at each of those different points didn't match this fellow. Now, when this was first happening, using this information to look at old cases, you know how DAs, all DAs these district attorneys care about is getting convictions. They're supposed to care about whether they've got the right guy or not, but pretty much they're biased. It's called confirmation bias. We know this is the guy that did it, and so they go and test this poor guy, and it's not him. And so the DA then comes along and says, well, this guy did it with another guy. Because they didn't want to lose a conviction. Also, they didn't want to admit that they put the wrong guy behind bars for 10 years. And that they're going to have to pay him a lot of money. Okay. So, in fact, all these different cold case files that, now remember, all these different results are in a big database now in state and federal crime databases. So what often happens is you've got these people who've been convicted of a crime because DNA was found at the crime, but at that time they didn't have the technology. They go back and they test it and they find out, oh, they've got all these people in jail who actually aren't guilty. But they find out that another person who had been in jail but is now out matches. So they realize not only did they have the wrong person, but they also know who the right person is who actually did these things. A lot of these individuals might have been on death row. Has anybody who's ever been put to death actually been not guilty? Almost certainly. So there's now something called the Innocence Project, looking at old cases. Now, obviously, if you don't have DNA, you can't do this, right? So there's a lot of old cases where they didn't have DNA. Those people are out of luck. So here's our DNA profile. A DNA profile is the number of repeats an individual has in both copies of all 13 STR markers. Like I said, 13, that's old. It's actually, there's more markers now, or they use these single nucleotide along the DNA, a bunch of those. But either way, originally it was the chances of someone sharing the exact same DNA profile with someone else with 13 were about 1 in a billion. That's not the case anymore. It's actually probably 1 in 30 billion. Remember, there's only around 7 billion people on the planet at this point. Now, the nice thing about this is you can actually eliminate suspects, because if you think about science, remember, we want to disprove hypotheses. Well, each suspect is a different hypothesis. So we can get rid of certain suspects because they don't match. We can say, oh, I think this guy did it. They collect a sample from him. They compare it to crime scene DNA, let's say semen, and they realize, oh, it's not him. So he's gone. He's free to go. They do it with all these other suspects, let's say, that they have, and they get a match. Now, there's still a possibility that the match is wrong. Now, when would someone share the exact same results at these 13 different regions as someone else? They share their DNA completely. Who shares, yeah?
Identical twins.
Huh?
Twins.
Twins, identical twins. Exactly. So, if you're an identical twin and you go and commit a crime, they can't convict you. Remember, there has to be reasonable doubt, or beyond reasonable doubt, that someone committed this crime. So all they can say is one of the two twins actually did it. But you have to be exact. You can't say, oh, it could be this twin, let's lock them both up. Besides, they have identical DNA, so they're probably all both guilty of at least something. And off they go. No, you can't do that. So with twins who actually go ahead and one of them commits a crime, essentially you cannot exclude, well, you could with other information. You could say, oh, we know for a fact that this twin, A, was at this party for this amount of time. And multiple people can actually testify to that, whereas no one knows where this other twin was. Of course, the defense could say, how do you know the twin that you think is the twin that was at this party was not the identical twin, since you can't tell them apart? Well, usually you can tell them apart. Now, that was the case for a while. It's different now. You can actually now tell differences among identical twins. Not from this exact data, but from other molecular data. So we can actually look at, because remember, we've got a mom and a dad. There are 4 possibilities in terms of these different STR bands. So let's look at mom. Here's mom, and here's mom, and here's dad, and here's dad. Okay. Here's baby number 1. We'll say baby number 1 gets this from mom and this from dad. Baby 2 gets this from dad, that's the lower one, and this same one from mom. Baby number 3 gets this from mom and this from dad. And baby number 4 gets this from dad and this from mom. Okay? Those are the 4 possibilities that we have. So here we are again. We can see that mom would share this and this, the same allele with these 2 babies, whereas dad would share either one of these. Okay, so here we are with dad again. Okay, and here we have mom again. So mom is either with this one or this one. And now we have mom is either this one or this one, and dad is either one of the 2, the lower one or the upper one. There are some things that aren't possible. So this baby has 3 regions. Why can't they have 3 regions? Remember, you get one from mom and one from dad. You can only have 2. Now, unless this is a baby that has a chromosome abnormality called trisomy, it's impossible. Similarly, can't have 4. Can only have 2. Now, it turns out sometimes there's these chromosome duplication events, but you don't have to know about this. This is just a little added information. So you might have, let's say, a plant that duplicated all its chromosomes. So the mom and the dad were each diploid, and there was a hybrid, and there was a duplication, and the babies now, instead of having 2, actually have 4. But you don't have to know that. There aren't any tetraploid humans out there. Okay. Okay. Let's see. Now remember, there is one thing I want to point out. You can have the same, if mom and dad both have the same band at the same place, that means the STR is exactly the same length, then you can have one place, let's say, if mom and dad actually shared this allele, then you could have a baby that had a double band at a certain place. Okay?